Hello everyone and welcome to Grand Mesa, where I'm coaching the ski team in the Colorado Mesa University Invitational. And as you can see, this is an absolutely beautiful place. Some of you may even be from around this area, not very far from Grand Junction. It is the incredible mesh of the desert and the ski country. So today I want to talk to you about something that's very pertinent to the outdoors and will excite many of you as we look at the growth requirements of different forms of life. And we're actually going to go ahead and sample in this beautiful area to see if we can grow organisms from the soil or the snow. I think I'm going to sample the snow nearby where the horses are. And actually, can you see? I'm going to show you around just a little bit more. If we look down below, you can see where all of the horses are. And we imagine that there's quite a lot of microbial diversity in this area, from canyons to the dude ranch here that we're staying on. So I want to talk to you today about the cultivation of bacteria. How do we take them from an environment like this that is so beautiful, and how do we then culture them in the laboratory environment and isolate them, see what's growing, what is the biodiversity in that area, with old-fashioned culturing methods. So I want to start by talking to you all a little bit about what organisms need in order to live. So what are the elements? If I were to bring a periodic table and say, what are the things that an organism needs in order to survive? So we would talk about what are those elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Or said another way, every living thing requires schnapps in order to grow. So schnapps: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Now, this is super interesting because at one point in time, and maybe some of you remember this, a scientist named Felisa Wolf-Simon was studying those microorganisms in the extreme environment of Mono Lake in California. She found an extremophile that appeared, at least based upon the work that she did, not to require phosphorus, which is crazy because we can recognize where some of these elements are found in macromolecules, right? Think about ATP, where phosphorus plays a central role. Think about the amino acids, for example, cysteine, that requires sulfur. We can think about all manner of areas in which we see carbon, hydrogen, nitrogen, and oxygen. For example, amino acids, we know that nitrogen is very important in the amino group and also often in the R groups. So thinking about the idea that one of these major elements, right, require ball life, that she said, well, I found a form of life that doesn't have phosphorus, but instead it uses arsenic. It replaces arsenic in all the places that you would see phosphate or a phosphorus. And guess what? She went under this extreme scrutiny by the entire, not just a nation's worth of scientists, but the entire world, because some of her procedures weren't rigorous enough to actually show that phosphorus had taken the place of arsenic in the macromolecules, but instead just that the organism managed somehow to live on a plate without phosphorus, but if arsenic was placed there, it would live. And so it was such a hoopla. So the major elements, we like the idea that all organisms need all of these. Now, they're not the only major elements. We know that there are other elements that are also required. For example, ions, metal ions. There are a lot of enzymes that can't do their catalysis without enzymes, iron being one of those. We know also that there are some organisms that are more resourceful than others. Some of them can live in very minimal or media. So when we sample the snow today, we're going to find that some organisms will be tough enough to live in the snow, that they will be tough enough to hang out there and not require a whole lot of added nutrients, whereas maybe some organisms would require that they live in a much richer, more filled with organic growth factors. So some organisms have to have pre-formed growth factors. They can't synthesize their own. And when I say growth factors, what I mean is nucleotides, amino acids, pre-formed building blocks for the macromolecules that we talked so much about in lecture. Now, note that these are all called major elements because they're required in substantive quantities in order to enable those biological processes of life, the synthesis of macromolecules. But there are also other elements that are required in very, very trace amounts. And so, fittingly, we call these the trace elements, or rather than calling them macronutrients, we call them micronutrients. These include some other iron ions, manganese, molybdenum, my favorite, zinc, cobalt, nickel, and copper. And there are things like cobalt that are required for the synthesis of vitamin B12. There are things like zinc that are required at the active site of enzymes. And for example, you're going to get so jazzed about this. The zinc ion is found at the center of the active site of an enzyme called alcohol dehydrogenase. Alcohol dehydrogenase is the enzyme that enables Saccharomyces cerevisiae, or yeast, to make alcohol. But catch this, it's also the enzyme that enables us to detoxify alcohol. So at the active site of that enzyme, we see a zinc ion, and I'll point it out to you in this image. So you can recognize that without zinc as a special helper, we call that a cofactor to an enzyme, it wouldn't be able to detoxify alcohol, or likewise in yeast to make alcohol. These ions are used in reaction catalysis. So let's turn our attention now to looking at the different kinds of media that we can use to grow organisms on in the lab. And we're going to start simple with our classification of media, looking first at this liquid versus solid classification. Now, this seems pretty simplistic, and in fact, already in Lab 1, you have worked with these two different types of media. However, the important distinction needs to be properly noted. That is that liquid medium, while we can enrich and grow up a lot in a liquid nutrient broth. Now, what I mean by enrich means if we want to make a lot more of a particular organism, say when I collect my sample here, I might put it into a liquid nutrient broth medium first before then putting it onto solid agar so that I can make more of whatever I want to look at. But you'll note that there's something very important that we cannot do in liquid broth that we can do on solid agar. Remember, looking at the single isolated colonies and that huge take-home message that a single isolated colony comes from one parent cell, we can take that and know that we have a pure culture. So the ability to derive a pure culture on our medium is made possible by solid agar. And it was tough for scientists to discover a medium, a solid medium that would enable this isolation. In fact, it was back in 1881 that Robert Koch, for the first time, used a solid medium. Here's what he did. He took a potato, threw it into boiling water, boiled it, cut it in half, and used the face of the potato to streak on. So this enabled him to get isolated colonies. That was crazy cool. And he was like, we've got to be able to figure out how to do this for all organisms, because of course, while things like yeast really, really love potato, not everything loves potato. So he went through many iterations of trying to find a solid medium. He even tried adding gelatin. In fact, it was a coworker of his that did that. And of course, what happened was they put it in the incubator at 37 degrees and it melted. So no go. So finally, they figured out if they could take an extract from Rhodophyta, or red algae, they could utilize the polysaccharides in that to make agar. And agar is wicked cool because it doesn't become liquid until very high temperatures. And it's solid at most. And so it enables us to have that really perfect solid growth surface on which we can streak for isolation. Our next media classification is defined versus undefined, or we could say simple versus complex. I like to think of a defined medium as being like the bread and water dinner, whereas a complex medium is like the 7-course dinner. So the more undefined, the more preformed growth factors, amino acids, nucleotides, vitamins, and those things required to make macromolecules. So a defined medium, if it's the bread and water diet, is only going to have just the bare minimum macro and micronutrients that are required for the synthesis of macromolecules. So a defined medium has to be very carefully made. We actually literally make it in the lab by weighing out every single little component that's in it. So if there's a certain amount of grams of glucose, we'll put that on the scale, weigh that out, and then we'll add the ammonium sulfate or the potassium monophosphate potassium phosphate dibasic. So we'll add each one of those components a little bit at a time, and we know exactly what's in it, right? There isn't any guessing as to what we put into that medium. So this is kind of expensive. As you can imagine, you have to purchase every single little component of the medium. It's also pretty complicated to prepare. You have to weigh out every single little component. So the upshot of this is that with this, we can define exactly what a microorganism needs. So if we take a sample here from on site and we put our organisms onto a defined medium, we can say that if they live, that's all they need for growth, right? The things that we put in there are all that they need in order to grow. So the more resourceful bacteria, they're going to be capable of growing on a defined medium, whereas bacteria that are a little bit less resourceful are going to require something that is richer, an undefined medium. So the defined medium, the bread and water diet that we're going to try for our organisms, and when we take our sample back to the Laramie lab, we're going to put it onto a GSA plate. GSA stands for glucose salt agar, meaning that in it is only glucose, that's the only organic, and then all of these inorganic salts. But notice how you can see where all of the major nutrients are coming from, such as the sulfur, the nitrogen, and the phosphorus. So these are the salts that are found in the medium, the ammonium sulfate, potassium potassium phosphate dibasic and monobasic, magnesium sulfate, and of course the glucose that we mentioned is the only organic component. So this is all that you find in GSA. It is defined, right? It is a minimal or simple medium, right? Very minimal, very little. And a bacterium that grows on this has to be very resourceful. It has to be able to build all of its own growth factors from just these few ingredients. Now, on the flip side, we could look at a much richer, more undefined medium. And the way that we make these is by taking an extract from another living organism. Remember how we talked about Louis Pasteur taking an extract from beef and how we contrasted that with an extract from hay? You can take extracts from lots of living things, but the interesting thing about doing that is that you actually can't tell what's in it. So if we were to take a chicken extract, right, there are chickens on this ranch as well. If we were to take a chicken extract and we were to put that into a medium, we would know that there was a lot of good stuff in it. We would be able to say that there were macromolecules, proteins, nucleic acids in their full form, right? But the nucleotides that comprised them. We could say that there were a lot of vitamins and there were a lot of essential ions. So we could say that there's a lot of stuff in there, but we can't tell you exactly what's in there. Because let's face it, that's going to depend partly on what the chicken ate. So we can't say exactly what's in an undefined medium. We can't use it to define precise growth requirements. We can only say that it's rich and that most things are going to like to grow on something like a TSA. So TSA, or tryptic soy agar, is what we're going to be working with in the lab. This contains a yeast extract and a soybean digest, and we're going to see what kinds of things love to grow on that rich medium. Now, I want you all to make a hypothesis right now. I want you to hypothesize from the sample that we're going to take from the snow next to the horses, hypothesize whether more organisms will grow when we put that sample onto a TSA or when we put that sample onto a GSA. And actually, you might even pause the podcast and write down that hypothesis now. Let's go ahead and talk about another category. This is selective versus differential. To understand selective, we're going to call on something that I have a feeling every one of you knows about. If you've ever had strep throat, or you know someone who's had strep throat, then you know that one of the things that we tell them to do is to gargle with salt water. The salt is actually selective against Streptococcus. So those organisms in the genus Streptococcus won't grow in the presence of that salt, whereas things in the genus Staphylococcus will grow just fine. So that's an interesting example of selective, right? Salt selects for staph and suppresses the growth of streps. So that's what we mean by selective. It selects for the growth of a desired organism by suppressing the growth of another organism. And that's a bit different than differential. A differential medium means that both organisms can grow on the agar, but some will utilize a compound in that agar that causes them to turn a color, or they cause that agar to turn a color. And then we can say, oh, well, that organism makes acid, clearly because it's turned the pH indicator in that plate to a different color. Whereas another organism might stay colorless and won't show that change. That's a differential medium. So selective, something dies, right? Something actually gets suppressed and can't grow. Whereas differential, we're just telling the difference between the two. We're going to put our sample here onto a medium that is both selective and differential, and in the lab you're going to do that as well. You're going to be working with eosine Y, methylene blue, or EMB agar, where you're going to add, you guessed it, eosine Y and methylene blue dyes. Those actually select against the growth of gram-positive bacteria. So gram-positives can't grow on this, whereas gram-negatives can. Interestingly, a lot of environmental microbes, like in our environment here, are actually gram-negative. So we're going to see that a lot will be able to grow on this, but there are also some gram-positives. Like remember in the soil when we talked about Bacillus subtilis, but also other species of Bacillus that can grow, and they're gram-positive. So we would select against those. So any sample that had gram-positives in it, the gram-positives would die, the gram-negatives would live. But check it, this medium is also differential. So the gram-negatives, we might be able to tell the difference between them. So for example, E. coli, we know that that is an enteric bacterium that lives in your gut. That's going to turn colors on this because it ferments lactose and it makes lactic acid. And that's going to cause it to turn a dark purple and have this really beautiful metallic sheen. So if we take our sample from our horse snow, we expect that some of that will be enteric, and we'll see, well, will we see some things that turn dark purple and have a metallic sheen, whereas other things will not turn dark purple. They'll just stay kind of the color, kind of a colorless, not remarkable in any way. So this is a selective and differential agar. So as we transition, I want to, I'll go ahead and show you the results as we streak that sample when we get back to Laramie. But I want to draw your attention to some ways that we talk about the classification of microorganisms based upon what they use as an energy source and what they use as a carbon source. So let's take a look at some of these classifications. So we'll begin with what we would think of as the primary producers, and these are going to be present in all environments. Primary producers are everywhere because they are at the bottom of the food chain. They are the food, or they are the food makers. So they take their energy from, in the case of photoautotrophs, they take their energy from light. And notice that the first part of the name in these descriptions always or photo, right, is referring to the lights, the energy source. Autotroph is referring to the fact that they're self-feeders. They're primary producers. But more expressly what it means is that these organisms can take inorganic carbon dioxide and they can convert it into an organic form of carbon. So they are making organics. Cool. We know that plants, right, trees are photoautotrophs, but a large number of bacteria are also photoautotrophs. So now, if we've got this strategy down, we can think about some other categories. Photo, light, energy, but some organisms don't get their energy from light. Some organisms get their energy from chemicals, like us. We get it from organic chemicals, but there are some organisms that are also primary producers that get their energy from inorganic chemicals. These are called chemoautotrophs. Notice again, the autotroph is there again. These are primary producers. Think about an environment not like this one. In fact, the light is starting to go down. But think about an environment that never gets any light at all. The primary producers, say deep down in a hydrothermal vent, they have to be chemoautotrophs because they can't use light as their energy source. They have to use chemicals. In this picture, we're showing an image of sulfur, elemental sulfur, right? Just sulfur. Some organisms look at that and they say, yeah, donuts, and they eat that as their energy source. So they utilize the inorganics in that sulfur as their donuts. So these are the primary producers of extreme environments, but I want to teach you another term, lithotroph. So sometimes these are termed chemolithoautotrophs. Lithotroph quite literally means rock eater. So rock eaters, right? These are chowing down, and this is such a beautiful picture of elemental sulfur. So while rock eaters are likely my favorite in all of the categories, I want to move on and talk just a little bit about a strange category, one that maybe you have trouble thinking of an example for, photoheterotrophs. So hang on a minute, we know photo means that the energy comes from light. However, heterotroph, now we're starting to talk about something that's not a self-feeder. That is that it eats other life or it eats organics. So this is kind of complicated because you're trying to think about something that would need light to excite electrons, but that would utilize an organic molecule as a source of those electrons. The purple sulfur bacteria are a great example of this. Have you ever been to a stagnant pond and seen a little bit of purple growth in it? These are going to start to develop in the Winogradsky columns that we're going to be building come next, it's 2 labs from now. So you're going to get to see some of the purple sulfur bacteria, but they use light to excite their electrons, but they strip those electrons off of organics. So this is a weird combination of a photoheterotroph. Most of the organisms that we work with in lab, and including us, fit into this last category of chemoorganoheterotroph. Our energy source is organics. Our food source, our carbon source, we eat other life. We are not self-feeders or primary producers. We eat other things. So organics serve as both our source of energy and our source of carbon. It's interesting to also note that that's what the bacteria that we work with in lab most commonly also do, right? We streak them on a medium. Even the minimal medium that we work with, GSA, has a source of organic carbon. So we're typically working with organisms that do utilize an energy source that is a carbon source, that is also their carbon source of organics. So that is interesting to note, and special then to think about the fact that later in the semester we'll be growing a Winogradsky column, and you'll be actually taking a sample from some environment, maybe going to a lake that you know of or a river that you know of, and you're going to be growing up some of these other interesting life forms in there. We need to leave this behind because we have the last challenge of the day. There are so many today. You need to be able to learn to aseptically pipette. You need to do the EMB, GSA, TSA inoculations. And you also, lastly, need to do dilution problems. But for the dilution problems part of our lecture, we're going to return to the indoors. The sun is starting to go down, and we're really going to start doing some math. Before all the light is gone, I snuck down here next to the horses, and we're going to sample some snow in this area, see what we can see, and hopefully get, I've got my little sample vial that I take along with me, so we're just sampling a bit of snow to see what we might find on our EMB, our GSA, and our TSA.